Welcome to our Unit 6 project on genetics. Our genetic disease is hemophilia. The inherited disease hemophilia is caused by a genetic mutation in genes that code for proteins that are essential for blood clotting, not allowing hemorrhages to clot. We're going to focus on hemophilia A as it is the most common type. It is caused by a deficiency in the gene factor F8, which is a clotting protein. This could be caused by mutation of this gene, which could be gene inversions, frameshift mutations, or deletions, usually occurring between sister chromatids during meiosis I. Meiosis is how this genetic disease is passed down from parent to offspring. This disorder is expressed in the X chromosome at position 28, which is a sex chromosome, making this disease X-linked. Recombination does not take place. This disease almost always occurs in boys and is passed from mother to son through one of the mother's genes in an X-linked recessive pattern. Most women with a defective gene are carriers, meaning they experience no signs or symptoms but can pass the genes on to their children. It is most expressed in boys because they only inherit one X chromosome, which comes from the mother, while girls have a healthy chromosome that can mask the effects of the diseased one. Some of the symptoms of the disorder include unexplained and excessive bleeding from cuts or injuries, many large or deep bruises, pain, swelling or tightness in your joints, blood in your urine or stool, nosebleeds without a known cause, and unexplained irritability in infants. Treatments include clotting factor concentrate, where the clotting factors from human plasma are separated. Others are cryoprecipitate, fresh frozen plasma, and desmopressin.
So our cancer we're going to be talking about is synovial sarcoma. So first, mitosis takes place, and what happens here is that cell division results in two daughter cells, each with the same number and type of chromosomes as their parent nucleus. There is the 6 phases, which happen to be interphase, prophase, prometaphase, metaphase, anaphase, telophase, and finally cytokinesis. So the cell cycle has 4 stages, which include G1, S, G2, and M. During G1, the growth and normal metabolic roles take place. Then in synthesis, DNA replication occurs. The final stage before mitosis is G2, where growth and preparation for division takes place. And finally, mitosis occurs. For growth factors, c-MET is a receptor tyrosine kinase encoded by the proto-oncogene MET, and it has a high affinity for hepatocyte growth factor. c-MET activation triggers cell growth, survival, invasion, migration, and also angiogenesis. Hepatocyte growth factor induces c-MET activation. Density-dependent inhibition is a process in which crowded cells stop dividing since the number of cells in an area force competition of nutrients and space. Anchorage dependence is a need for contact to a stable surface by some cells in order for them to grow in cell cultures, and it is necessary for cells to spread or divide. Transformation is a conversion of normal cells into tumor cells involving the changes of activity of number of distinct different genes and proteins in a cell. What happens is that mutation inactivates tumor suppressor genes, which leads to cells proliferating. After, the mutation inactivates DNA repair gene, and mutations of proto-oncogene creates an oncogene, which leads to mutation in activating several more tumor suppressor genes, causing cancer. The cause of synovial sarcoma is associated to be between the chromosomal translocation of chromosome 18 and X. It is unknown whether the mutation occurs randomly or follows a specific change of events, although it is known that it contains a mutant gene which contributes to the development of the disease. There is 2 different types of tumors, one being benign and the other malignant. The major differences between benign and malignant are that benign are non-harmful and they usually do not metastasize. For malignant, they are able to invade surrounding tissues and organs and also cause fatal damage. Synovial sarcoma happens to be a malignant tumor. For treatment for synovial sarcoma, you can get surgery removing the entire tumor plus the tissue around it, radiotherapy, which may be used before and after to reduce the risk of leaving cancer cells behind, and even chemotherapy, which might be recommended especially for advanced or metastatic cases. For the prognosis, we have survival rates of 50 to 60% at 5 years, 40 to 50% at 10 years, and it is higher if you are a female, younger than 50 years old, and when the cancer is located on your hand or foot. It happens to be lower if the tumor is larger than 5 centimeters and also if a reoccurrence happens after 69 months.
Thanks for watching!